or physicists or okay, yeah.
It's a wide background.
Now I usually give talks to sort of general audience, so some of the slides will, to an astrobiologist, be a bit sort of overkill. But if there are people with sort of no heavy knowledge of astrobiology, I think it's good to still have them there. Yeah. So I'm going to talk about a project called AMAZE. It's always nice to have a good acronym. AMAZE stands for Arctic Mars Analog Svalbard Expedition, and it's a project where we use Mars analog sites on Svalbard, which are these islands way north of Norway, 80 degrees north. Use these Mars analog sites to look at, well, basic science in terms of organosynthesis, extremophiles, you know, what sort of organic chemistry, what sort of life can you have in a sort of Mars-like environment. But even more importantly, we use those sites to test instruments that are either on the drawing board or are actually going to Mars on board future missions. And I'll show, well, it will be a sort of a mixed bag of this, that, and the other. Now, yeah, this is a light something. And the singing lights, maybe you, pitch dark. Well, not pitch dark. You can get those up, better. Good. It's always important to be properly dressed in the field. So this is part of the group of people that makes up the AMAZE project. Now, I'm associated with this center at the University of Oslo, and I run this project. But most of the people on it, and I think the most important collaborator we have is the Carnegie Institute in Washington, with this long-haired guy here called Andrew Steele. He's a microbiologist / astrobiologist. They constitute a major part of the project. And then we have people from NASA and JPL who make the hardware that's going to Mars and a lot of the instruments that we're testing. And of course, the Norwegian Space Center is my most important ally in Norway, getting at least moral support. Starting new disciplines, especially interdisciplinary ones, is not easy, but it's great fun. And not too long ago, I stopped calling myself a geologist and came out of the closet as an astrobiologist, which is for psychology and comments. But I still think I'm an astrobiologist. So. And then we have company, industry sponsors. This is my company. And then we have the University of Leeds is on board, and the Lunar and Planetary Institute. A group from Penn State working on weathering. And of course we have important collaboration with UNIS, which is the University Center on Svalbard. And the questions we deal with, which of course are familiar to an astrobiologist, but maybe not to the more common man, is, I mean, can you come up with any better science questions to spend your life digging into? I don't think so. Where do we all come from? How does life start? Can we find life elsewhere in the universe? And what's the future of life on Earth? This third point it's on the astrobiology sort of web page. And I must admit that I'm not too concerned about the future and the science I'm doing here. I'm mainly just into what's going on now and maybe going on elsewhere. And find a motivation for working with this is at least not difficult for me. Like Isaac Asimov said it, he was originally I think he was a biologist turned science writer. There are two possibilities, maybe we're alone, maybe we're not. Both are equally frightening. So, now when we start to try to figure out how life formed, where can we find life? I mean, you start to look for the building blocks, and that's organic chemistry. And through the later years, it's become evident that there are building blocks for life in the sense of simple organic molecules, more or less everywhere in the universe. You can see it in spectra from distant planetary disks. You can find it in interplanetary dust particles and meteorites. And building blocks of life, organic chemistry, appears to be present almost everywhere. Now, a lot of the hard knowledge about the chemistry from outer space, of course, comes with meteorites. There are many famous meteorites. This is one of them. This guy looks unhappy here, but he became much happier when he sent his car on a world tour on display. That's the first vehicle normally hit by a meteorite. One of the more famous ones for people working on organic chemistry and astrobiology is the Murchison meteorite, which fell in Australia. It's always good to see meteorites fall, because then they can pick them up immediately before they get contaminated by kangaroos, for example, or bush. And this Murchison meteorite revealed some startling surprises when it came to organic chemistry, in the sense that it had very complex molecules, stuff like amino acids, and things that previously were thought to be linked to primarily to biological activity. This rock has samples of that evidently formed by some organic synthesis type process in outer space. Then, of course, people hear about astrobiology and searching for life in the universe. They usually think about fairly complex organisms that you can communicate with. But before we start to look for those, it's much easier to start to look for something simple. And actually, before we start to look for something on other planets and design instruments to go to other planets, it could be a good idea to actually figure out if we can decide when life started on Earth. And that is not a straightforward question. As I guess we all know in this room, the planets formed about a little more than 4.5 million years ago. To start with, the Earth was a pretty hostile place. It was probably heated by meteorite bombardment, but at the same time the sun was very faint, so it could also have been very cold, and actually we don't know. But there was not much of an atmosphere, and more or less something to find in the basement of a barn, methane and ammonia. Then, after some time, volatiles that were sort of fixed within the Earth became released through volcanic activity, and we got nitrogen, CO2, and water. And at some stage, conditions became such that water was stable, and it actually started to rain. And that was extremely important, because then we got processes that, like erosion, you got rivers, lakes, and conditions that we certainly think are necessary for life. All life that we know on Earth requires water. But still the questions remain: when did it form on Earth? How do we detect fossil life? And what is true evidence for past life? What are true biomarkers? Can we use carbon isotopes, organic chemistry, or biogenic patterns, shapes? This third point is probably the most debated, because a lot of like in the Allen Hills meteorite, for example, where you have these strange shapes, a lot of, let's say, heated debate has started with looking at shapes that look very lively, but you can't use billions of dollars to send something to Mars and find a shape that looks lively and then conclude that there is life outside our own planet. Now, obviously, if you find something like this is a creature that was discovered this year, actually, at some kilometers water depth around a hydrothermal vent. It's like an albino lobster with fur. There wouldn't be much doubt that this is some new form of life. So, if you ever find something like that with a camera on a space rover, that's fine. Or stuff like this. I mean, that's a fairly convincing biogenic shape. If you find something fossilized that looks like this, I don't think there will be much argument whether we've actually detected fossil life. Now, interestingly, a lot of the so-called evidence for the first life on Earth is based on, I dare to say, it's almost gut feeling and looking at shapes. This is a very famous set of samples from the Apex Church in Australia. It's a very old sedimentary rock. And they were described sort of microscopic tube-like features, which were carefully drawn up and given Latin names and divided into families of single-celled organisms. Now, unfortunately, if you look closer at that rock, and especially if you use detailed Raman spectroscopy, you'll find out that it's extremely difficult to prove that this was life. And actually, it looks more or less like something deposited on a crack. And I'm sure, I mean, this will probably be debated for many years, but this is nothing we can take as evidence for life. Shapes like that, it doesn't hold. And of course, if you come into organic chemistry, if you find something like a piece of a cell membrane, yeah, you're getting into shape, you know. These things are associated with cells, and we really don't know how to make them unless we have life. Then you can look at the individual constituents of membranes, like lipids. If you find lipids, is that evidence for life? Well, it's not really good to say. And if you go down to amino acids, it's certainly not, because we know that amino acids form in the outside our own planet and can be found in many other places where they're not formed by life. And the problem is that we really don't know how life forms. And we have a lot of, you know, organic molecules and building blocks here, and we have observed life here. But it's the miracle part that's a bit troubling. Now, a lot of good science questions were formulated around this particular rock here. This is the Allen Hills meteorite, which was discovered in Allen Hills in Antarctica in 1984. The first meteorite they picked up that year, that's because it's number one. It's a strange meteorite because it's really not your typical kind of rock falling from the sky. It's a piece of a lava flow. So it's coming from a planet with active volcanism at some stage. And it was curated with great interest because these kinds of meteorites are rare. And then in 86, I think it was, 96, it was discovered that when this rock was heated, it released gases that were identical to those measured on Mars by the Viking landers. It had noble isotope or noble gas ratios like argon, xenon, krypton that were unique, different from anything we see on Earth, and the only place we know that has that kind of atmospheric signature is Mars. And then secondly, if you do simple calculations about the chance of a rock being torn loose from a planet and then being captured by the Earth's gravity field, there are only a handful of planets that are likely to deliver such material. It's the Moon, of course, and it's Mars, and perhaps Mercury and Venus. And through sort of various lines of reasoning, we're pretty sure that this rock is actually from Mars, and that was the first Martian meteorite that was sort of claimed to be from Mars. Of course, that spurred an enormous interest in finding out about anything possible about that rock. It was the first sample from Mars. Now you can use various radioactive clocks and cosmic ray exposure ratios and stuff to date the history. And this lava flow apparently solidified around 3.9 billion years ago on Mars. That's just after the planet formed. It sat around on Mars for quite some time until it was hit by something hit Mars around 16 million years ago. So it floated out around in space for about 16 million years and then landed on the South Pole, or close to the South Pole, for about 60,000 years ago. That's quite a story for a small rock. Now, and then of course there was a race to find the first evidence for anything unusual in a rock from Mars. And the first thing that was discovered were these odd carbonate globules. This is about 1/10 of a millimeter, 100 microns, which were composed of magnesium carbonate, magnesite, and dolomite, calcium magnesium. They were round, they had an interesting shape. Nobody had really seen anything like that before. And it became clear that somehow this formed by interaction with water with this rock on Mars. And then of course people got really interested because water and Mars and rocks and reactions and carbonates, they started to jingle a bell that sounds like maybe there's life here. And then they put it on the SEM and they found these strange shapes which looked awfully lively. You can almost see it move. And the group that worked in particular on these two aspects of that rock, they concluded that this, and also that it had also organic chemistry. You can find polyaromatic hydrocarbons of 6 rings. That we had organic chemistry there too, and there were also some magnetite grains that were a bit strange in shape that reminded of magnetite grains from bacteria. So anyway, they concluded, the group led by McKay, sitting here, that this meant that, well, each evidence by itself wasn't conclusive, but all these strange things in that rock, they chose to conclude that it had once contained life on Mars. They submitted that story to, well, they tried to submit it several times, and got it accepted finally by Science. And McKay went to Golding, who was the administrator of NASA at that time, and said, yay, we've got life on Mars accepted in Science, shall we hold a press conference? And Golding said that no, he thought it was a bit speculative, so he'd rather play it a bit cool. And then the tabloid paper, The Sun, in the UK, got hold of the story, and they used the biggest font they could find, to put life on Mars on the front page, and they called Golding and asked, now aren't you going to hold a press conference? And he gave in. So this is from the press conference, and it virtually exploded. Of course, media loved it. Some other scientists hated it. Bill Clinton went on TV and said it was the greatest scientific discovery in the last century or something. Just by chance, this came up whilst the Congress was deciding on the Mars exploration budget for the next 5 years, which by some strange reason got doubled. And, well, you know, off it went. And then NASA, of course, diverted a lot of the researchers to actually work on that one rock. And not all of them liked it, and a lot of them did not like the conclusions drawn from those particular shapes. And that's why I say you get up with some really good science questions when you end up with a story like that. How do you detect life? And of course, you can't use shapes to decide life, unless you come up with a unique shape that only can be formed by life, like a albino lobster with hair or a big dinosaur fossil. Right? So how do we detect life? That's not easy. And interestingly, they found out how you make these shapes. It's very simple. If you have, I mean, when you put a sample for SEM, you have to coat it with something that can get rid of the electrons. So the sample is gold coated. If you have a small impurity on your grain and you coat it with gold, the gold will build up around that impurity, and you will get shapes like that. So it's basically a gold-coated piece of something. Now, back in 96, 97, they of course tried to, with those methods available then, to find any microorganism in Allen Hills, and they couldn't. Now, 10 years later, we've found a lot of microorganisms in Allen Hills. It's full of microbes, terrestrial microbes that will move into any rock that lands on Earth to start to eat it. So there's lots of life in Allen Hills, but it's all from Earth. Now, back to our own planet. There are some sort of crucial cases which have been used to pinpoint the emergence of life on Earth sort of between 3.5 and 4 billion years ago. The oldest rocks on Earth occur in Iswa on Greenland. They are sedimentary rocks and some volcanics. And the sedimentary rocks have graphite, which you see as dark specks here. And of course, graphite, carbon starts to ring a bell if you really want to find evidence for life. And there has been done some isotope studies on this graphite that shows that it's unusually light. And we used to think that light, isotopically light carbon, is only made by photosynthetic organisms, which has turned out to be wrong, unfortunately. I'll return to that. But that graphite and the light isotopes has been used to as evidence for sort of traces of life or biomarkers in the Iswa rock. It doesn't hold. Then, of course, you have these famous called banded iron formations, which is essentially rocks which suddenly show up with lots of Fe3. And to get that, you have to have a high oxygen pressure. And we know that the early atmosphere of the Earth probably had no oxygen at all. And that at some stage down here you start to increase oxygen in the atmosphere, and that's the best way to explain that is photosynthetic organisms. And again, this presence of sort of rust, so to speak, at that stage is taken as indirect evidence that you're getting O2 from photosynthesis. Could be, but again, you can't send something to Mars and decide on the basis of that. Then, there's another example with apologies to my good friend Harald Furnes in Bergen, but this is a pillow basalt from South Africa, a microscope image of some strange tubular features which look like burrows in volcanic glass today, where microbes actually eat their way into the glass. Texturally, it looks similar, but this rock is turned into serpentine and whatnot. And you know, if you start to hunt with a microscope in any rock, you will find strange wormy features, which are you can't really use that to decide whether they once contain life. There is carbon in these things, even probably organic carbon, but again, you don't know when that carbon came into the rock, and you really don't know if that carbon was formed by life or some other abiogenic process. And of course, you have the famous guys in the Apex Church, which end up in the same debate. Then you have what we call the stromatolites, which is certainly a much more interesting case. Because stromatolites form today as algal mass, or bacteria and algae precipitate carbonates in shallow water. You get alternating layers of biofilms and carbonate, and they build up these characteristic mounds. Stromatolite actually means bouldy rock in Greek. They build up this characteristic pattern as they grow. And the only way well, it's actually not true, but we think that the only way to make this is by these algal mats. And that was until some bright guys at MIT decided to test that. And they took simply spray paint and they sprayed paint on a surface. They actually used a robot because they had to do it for a long time. Repeatedly, just spray, let it dry, spray, let it dry, and they got the same texture. So it's, you know, I think the stromatolites are actually formed by life, but how do you decide? Unless you start to look for biomarkers or actually organic chemistry in them as well. But to look for organic chemistry in 3.5 billion year old rocks, again, it's not straightforward because you don't know when those chemicals came into the rock. And on Svalbard, one of the places we're working at is a stromatolite site. They're 800 million years old, and we are trying to analyze those for biomarkers, but we found out that the biggest problem we had was actually to take a sample without contaminating it, and not at least to take a sample without getting biomaterial that was actually coming in on micro cracks. So it's not straightforward. But probably the stromatolites are the best evidence for life on Earth, and the earliest stromatolites are found around 3.5 billion years ago. And of course, there are various nice shapes in sediments that are certainly not easy to discern. These guys here are from a site called the British Springs Church. Now, shapes are nice, but in addition, they also contain organic chemicals like pigments, and I think also cholesterol, which certainly starts to ring more bells, and these are probably accurately described as fossil life. Then, down to about here, the only evidence for life on Earth we have is single-celled organisms. Then something went very wrong, and the Earth froze down through 3 periods, called the Snowball Earth period. So we had glaciers all around the Earth. And when that ended, we got the first multicellular organisms. Now, of course, these guys make fossils that are much more easy to decide actually look like life. And then, not too long ago, man appeared and has been debating these issues ever since. It is not staged, it is taken outside the archaeological conference in the US. Now, ever since man has been thinking about where he comes from and stuff like that, people have been speculating about life in the universe. One of the most famous guys is Giordano Bruno, who was a close friend of Galileo, who did the outrageous thing of suggesting that the Earth went around the Sun and not vice versa. And Giordano also concluded that all the stars out there were suns like our own, with planets around them, and that these planets also had life. He was burned at the stake, apparently not because of it, but because he was a very quarrelly guy. And Galileo changed his mind and survived. And then back to Mars. These, ever since we got good telescopes, and not the least the latest years with all these various Mars missions, Mars has become really a hot topic. And there are many reasons for that, not the least that Mars actually has active geological processes. It has volcanoes, like Olympus Mons, which is nearly 30 kilometers tall, and is probably still active, and has been active for over 3 billion years, sitting in one place. And other large volcanoes, you have this fantastic Valles Marineris crack, so to speak, or canyon in Mars, which is as long as the US is wide. And you have other features, very visible here probably, that looks like meandering rivers and remnants of lakes and stuff like that. And you have ice, CO2 ice, and also water ice on the poles. So there's a story with Mars, with heating, volcanism, ice, water, that becomes pretty interesting when you try to decide where to look for life elsewhere. The fun thing about working with other planets and meteorites is that there are lots of imaginative people working on those subjects, and it's a much more, how can I say it, it's a much more colorful environment than, let's say, classical physics, where you use stringent, do math and stuff like that. People who look out, they tend to really use their imagination. And one of the first guys who really studied the surface of Mars, Percival Lovell, start of the 20th century, he saw these stripes and features and channels and stuff like that, and he concluded that they were part of an irrigation system, actually, built by intelligent beings. Unfortunately, that didn't hold. But in the mid-70s, the first space probes, the first space missions that actually landed on Mars. Sorry about that. I'm going to turn it off. Oh, come on. There you go. There were two missions that landed on Mars, Viking 1 and 2. And they had with them, of course, a camera to photograph the surface of Mars. But more importantly, they had an experiment where they seeded Martian soil with a particular substance that would, if any microbes were present, they would, what should I say, eat that stuff and release a labeled marker, and you can detect that there were active microorganisms. The results from this experiment was unclear, and it's still debated today actually. The downside of it is that the detection sensitivity was very low. And it turns out that for that experiment to be 100% successful, you would have to have about as much, should I say, biomass per unit as cow dung. And it would only show that there's no cow dung on Mars. The stuff we have, the instruments we have today are much more sensitive. Then we have the Mars Pathfinder, who didn't have any instruments to look for life or organic chemistry, but it had a camera and it had some spectrometers which could look at rock chemistry and mineral data. And then, of course, you have the famous Mars exploration rovers, which were actually designed to work for 3 months or something like that, but they landed in early 2004 and they're still ticking, and have delivered fantastic data, again with no instruments that are designed to detect organic chemistry or even life, but lots of data on outcrops. It photographed the first sedimentary sequences on Mars, like these ones in the Gusev crater, which probably was filled with water once and had a sort of channel flowing out of it. These sediments are probably very, very old, so we don't know, but they might be as old as 3 million years. Interestingly, they contain salts like gypsum and chloride. And the only analog we have to that on Earth is actually sediment deposited in an evaporating sea. And one way of explaining this is actually, once upon a time, Mars had conditions where water was not only stable at the surface, but where you actually had evaporating lakes or shallow seas. Then there's another NASA mission on the drawing board. Well, it's actually further than the drawing board, because it's flying in 2009, which has loads of instruments on board. It's called the Mars Science Laboratory. And those instruments, including a GCMS and LIBS, Raman laser, and XRD, XRF, lots of stuff. Some of them are equipped to detect organic chemistry and to analyze for stuff like carbon isotopes and organic molecule masses and stuff like that. And this mission will have the possibility not to detect life really, but to detect the building blocks for life. And while the Mars exploration rovers are about this big, this is the size of a small field, but it's pretty sizable. It should carry 80 kilos of instrument payload. And there's another mission been worked up by ESA called ExoMars, which has a similar goal and similar instruments, but unfortunately it only can only carry 8 kilos of instruments. But it's scheduled for 2011, the last I heard. Now, because the Mars Science Laboratory is much heavier than the Mars exploration rovers, they can't land it the same way. I mean, the MER rovers are basically, they come into the atmosphere, they are slowed down by parachutes and then released with this cushion of pillows around, and they bounce along. That won't work with this thing. So they designed something brand new, and I bet you they're going to hold their breath when they land this thing. It's called a sky crane, where actually they have this thing that is, it's stopped by a parachute, and then it fires rockets, and this little robot is hanging underneath it, and it stands there in midair, and it lowers the robot down in wires, and off it goes. I hope that works. But in any case, this thing has a very sophisticated set of instruments, and not least a sample handling system. And the 3 aspects of this rover that we're testing on this AMAZE project. It's the instrument called the Chemin, which is a combined XRD-XRF, which is about, the field version that we had this summer is about that size. The flight version is a lot smaller. And it's a portable GCMS, gas chromatographer, mass spectrometer. And the world's smallest rock crusher is the size of a thermos. It looks like a normal jaw crusher, and it works on a 6-volt. We can make it run with a 6-volt battery. It's really freaky. It has a big spring, like you see in old-time watches, and this battery will wind that spring up during something like 6 to 8 hours, and eventually the pressure becomes so enormous that a small rock is turned totally into powder. So it actually powders a rock with the equivalent of a 6-volt battery. This thing will probably have, it's debated still probably, I think, but you will have to have a nuclear source to have power rather than a solar panel, because you need so much more energy. Yeah, so back to AMAZE. Now, we have our favorite site up on Svalbard, in this area here, which is where we started to work. It's called the Bockfjord Volcanic Complex. There are other sites as well, but I'll mainly talk about this one today. It's an area where you have, of course, glaciers and permafrost. You have 400 meters thickness of the permafrost. Still, you have hot springs coming up through that permafrost, and you have volcanoes. This is one of them. There's 4 or 5 of them. That's sitting in this area, and this interplay between volcanism, hydrothermal activity, permafrost, ice, is something that's reminiscent of Mars. So this is our main study site. It's called the Sverker volcano. It's the erosional remnant of something that was much bigger once. It's been run over by several glaciers. It has the remnants of a shoreline about 300 meters above, or 200 meters above sea level. And it has pillow lavas, which are evidence of lava flows running into water, all the way to the top. And actually there are other volcanic centers in this area sitting at 1,000 meters altitude, which still have pillow lava. And the best way to explain these observations is that the Sverker volcano erupted through a thick ice sheet, or actually all these volcanoes did. And that ice sheet was probably around 1,000 meters thick, meaning that you're looking at much colder ambient conditions on Svalbard than today. If you play around with the climatic models, that would indicate a sort of mean annual temperature of minus 30 degrees C. It's not like Mars, but it's pretty cold. The only age constraints we have on these eruptions is some argon-argon data, which suggests around 1 million years old, but still the data are not exceptionally good. But the interesting thing about this volcano and the other ones is that they are totally loaded with these carbonate globules that are identical to the ones in Allen Hills. And when I saw Allen Hills published, I thought, well, I've seen that before. I'm sure there's stuff like that everywhere in the world, but nothing surfaced. And then the people working on Allen Hills, not the McKay group, but the people that tried to disprove him, they found my paper describing these, and I started to work with them on the Allen Hills meteorite, and they got samples from Sverige back in the late 90s. And that's where this link between Mars research and the Bockfjord volcanic complex started. And of course, it's not easy to have a small rock that's traveled, millions of kilometers through space from another planet and decide what happened. Here you can actually take labs, instruments, scientists, whatever, to the area and play around like, in Norway we have this program called CSI Miami. Do you have that in Sweden? It's sort of a crime scene investigation thing. Here you can really go in and investigate the whole area to find the context of these carbonates. So our first expedition up there was in 2003. I persuaded the university to take responsibility to charter an icebreaker, and then I sold out the slots on board to a network of people. I knew some of them from beforehand, and just word went around, and it was not difficult actually to fill that boat. It with a little less than 20 researchers. So what we do, I first go through the instruments and then some of the science results. So what we test up there is a suite of different technologies that are either on its way to Mars or at least on the drawing board. And we try to act as if we are a rover. So we go into an area, and to look for life, the first thing we use is our eyes, like the camera on the rover, to look for interesting stuff, like geologists do. So we look for somewhere to focus. And then we go in we use fluorescence, we use lasers. Now, lasers have the ability to, if you have a certain wavelength of light, it will excite fluorescence from organic chemistry. This is like, in the old days when I was still going to discos, we had this blue light, you can see the dandruff on your collar. It's the same thing. We're looking for dandruff. So these lasers are designed with different wavelengths to pick up different organic chemicals. This is the same patch on a rock, showing various types of organic chemistry excited by different wavelengths of laser light. Then once we find something that gives response on the laser, we go in and we actually sample it and do more conventional chemical methods. One of them is to use protein microarrays, which is essentially, you can print on a glass slide, used on the standard microscope, you can print a lot of questions, yes-no questions, designed to detect a particular chemical substance. And once the substance is present and reacts to that chemical test, it will fluoresce on the laser light again. So all these things lighting up, that's the answer yes. And you can design it to test for cats, reindeer hormones, cell walls, DNA, anything. This is instruments being developed by the medical industry, but it's also being sort of broken into the questions that we work on for Search for Life. We basically use this to test actually a lot of the other instruments, whether they're sterile before we start to sample, because it's the fastest way to detect contamination. But we also use it to look for certain biomarkers like pigments like chlorophyll and stuff in the field. Then we do PCR, which is taking a particular sequence of DNA and sequencing it. And what's most usually PCR runs are designed to detect a particular species of bacteria. We've designed ours to detect particular types of what we call active groups on the DNA that tells us whether the organism is oxidizing methane, whether it's eating sulfur, whether it's photosynthetic and stuff like that, because we're interested in what kind of chemical process these various microbes are hitchhiking on when they get their energy. And of course we do all this in the field. We become better after some years. The first year, one of us had to sit on a box with his hands inside the glove box for a little more than 6 hours to get the run through. It was very cold, but it worked. Then we use something called ATP luminometry. In any living cell, we will have mitochondria. That's sort of the power station of the cell, which contains ATP, which can store or give off energy. An active ATP will emit a certain frequency of light, actually photons, and you can detect these photons and actually measure in any sample the presence of ATP activity and convert that to more or less a number of cells. This is one example from some of these carbonate terraces in front of the hot springs, which look totally dead. But they're loaded with microorganisms that live just underneath the surface. They don't like UV radiation, so they need sunglasses. So they use the carbonate minerals as sunglasses, so to speak, and they live just beneath the surface. So if you crack open the rock, you will find this green layer about half a centimeter down. Then we use more sort of odd stuff like the horseshoe crab. The horseshoe crab has the most effective immune system known to any organism. And there's actually a whole industry that captures horseshoe crabs and drain them for blood. And take that blood and extract a certain chemical substance called LAL. And LAL has the ability to detect cell walls. So that crab is extremely efficient in detecting any foreign microorganisms invading itself, in the sense that it can detect cell walls. And this instrument is so sensitive that it can actually detect the cell wall from one single microorganism. The first instrument we brought up there was pretty big. Now it's reduced to that size. And I think this one actually was sent to the International Space Station about a week ago to be used to detect, let's say, contamination and stuff on board the space station. Then we have some of the instruments from this Mars Science Laboratory, like the infrared thing. And this year we also had with us a so-called cliff-bot. It's a rover that's designed to enter down into a crater. I mean, instead of drilling down into the surface, you can investigate down into Mars by landing on side of the big crater and then going down the crater walls. This is actually 3 robots working as a team. These 2 guys here, which is essentially a reel with a fishing line, but they're movable and they will give out line or hold in line as you give it commands. And you have this guy which is hooked up to those and can traverse down into actually almost vertical terrain. It's equipped with a camera, of course, to see where it goes, and with a sampling device very similar to with a rock abrasion tool, a RAT, very similar to what's on the Mars exploration rovers. This is a prototype version where there are not many other instruments on board. It's not been, what should I say, okayed for Mars flight yet, but it's a robotic platform that we're testing in connection with these instruments. Then we've also started to test sort of human spaceflight technology, like the latest fashion in spacesuits. The scenery on Svalbard, especially with these red Devonian rocks, are very Martian, and you get some good photos when you put an astronaut into that. But more important, we test how an astronaut can use these instruments that we bring along. For example, a hammer as an instrument. But more sophisticated stuff like a handheld Raman laser. And yeah. So by combining all these different instruments, I mean, there's not one single instrument that's going to detect life on Mars. You will have to detect it by different independent methods. And we managed to put together a suite of instruments that could, with a high confidence level, detect down to one single microbe. So if we're able to ruggedize these things and send them to Mars, we can detect one single microbe if there is life on Mars and if that life looks like the life on Earth. But more importantly, you have to send the stuff up there without bringing any contamination from Earth. And that's maybe a bigger problem, because with the Mars Science Laboratory, they now have to actually sterilize the whole space probe. And they've never done that before. And that's going to be quite a challenge. And when it comes to sending stuff up there to actually detect microbes, it's going to be even worse. Now back to some of the slides. In particular, the first thing we sort of dived into was these carbonates. Now, when I worked there as a geologist many years back, I was looking at volcanic rocks, and anything that were, looked like alteration and carbonates and stuff like that, and clay, I just thought, well, I didn't want to deal with that. So I had been in that volcano many times, but I'd never noticed the carbonates, except these guys in the thin section. And we went there, and we found that there were lots of strange breccias, like just volcanic rubble that was cemented together with magnesium carbonates. And of course, they were associated with these guys here, which are sitting in the vesicles in the lava, and they're actually sitting all over the place, anywhere they can. And so, one of our first questions were to find out how did the carbonates form. And then we sort of laughed a bit, and they said, yeah, maybe they're even associated with microbial activity. And what they found is that, I'll go 2 slides down. First of all, the stable isotope data on the carbonates were a bit surprising, because they actually showed very light oxygen, indicating glacial meltwater. And the only other carbonates on Earth with that extreme oxygen composition are carbonates that form by freezing of glacial meltwater. So usually you think about carbonate formation by sort of some form of evaporation, but you can concentrate and degas water just as efficiently by freezing than you can by evaporation. So the carbonate isotope ratio, oxygen, of these carbonate deposits in the volcano suggests glacial meltwater and possibly freezing. And if you start to look at the chemistry of these things now, they're very magnesium rich, and like in the Allen Hills meteorite and elsewhere, when you find magnesium carbonates in a volcanic rock, you immediately think about alteration, that you dissolve magnesium rich minerals. But it turns out that when you freeze normal meteoric water with a low magnesium calcium ratio, as you precipitate carbonates, you will drive that fluid into, or the residual water into more and more magnesium rich compositions, and pretty rapidly you will start to precipitate dolomite and magnesite. And that was initially a theoretical constraint. But if you look at actually glacial meltwater on Svalbard today, this is typical meteoric water in terms of bicarbonate, sulfate, and magnesium calcium. It sits up here, and if you start to freeze that water and precipitate out minerals, you drive it down here, and you will soon end up in this region where you start to form the kind of carbonates we see inside it. So the best way to explain this predominance of magnesium carbonates inside it is probably that the volcano, as we've seen from field data, erupted through ice, glacial ice, melted it, and it was soaking wet until someone switched off the heat machine. And if you had sort of minus 30 degrees average temperatures, it probably froze solid, and all this meltwater precipitated out carbonates. That's the best way to explain all the data. And if that's the case, that also explains why you have these similar carbonate globules in the Martian meteorite Allen Hills. That something happened on Mars at that stage where water froze. And things freezing on Mars is a fairly straightforward scenario. Then we started to look in more detail on these carbonate globules, and to our surprise, they were full of organic material. You could detect that by Raman. Yeah. So you have a core of actually iron oxides and hematite, magnetite. Then you have these magnesium carbonates sold out here, and then in this sort of brown layer here, 2 layers, you have carbon, and it's not graphite, it's actually organic carbon of some sort. And we've done studies the last month that suggest that it's PAHs, these polyaromatic hydrocarbons, this benzene ring type structure that's sitting in this setting, and probably also other types of organic chemistry. We also found these things enclosed within minerals. And in that case, they're not formed by glacial meltwater, but they're actually formed from fluid inclusions inside mantle minerals, for example. And they form the same carbonate globules, apparently, probably because they freeze. And these things, which are totally encapsulated, also have the same organic carbon content. And interestingly, that organic carbon, which is certainly formed abiogenically, is extremely light. So this example and others actually that have been published shows that you can get very light carbon without having photosynthesis. But this is actually an interesting way to form the building blocks of life. We don't know the exact process, but something happens when you freeze these probably hydrous fluids down and dump out carbonates. You also dump out organic chemistry. And if you look at a very strange resolution. Oh well. Well, this is a close-up of one of these vesicles in the basalt, which is coated with these red-looking globules. And you see all this gray stuff here. It's a biofilm. It's some form of organic chemistry. Actually, it looks like keratin. For those of you familiar with that. And the globules themselves are coated with biological or hydrocarbons in some form. And that image, which doesn't show up at all, I can, if I do like this. This is a sample of these globules, which is stained with I forgot the name of the stain. It's a stain that attaches to DNA, so you can analyze by fluorescence the presence of bacteria. And it turns out that these globules are teeming with life. They're microorganisms that are apparently living off this organic carbon that's formed here. So where these globules are, those microorganisms move in and they actually eat them. This doesn't mean that there are similar organisms that are present in the Allen Hills meteorite or on Mars, but it certainly raises the question whether you have similar organic chemicals in the Allen Hills meteorite. So we went and looked for that, and lo and behold, we found it. So this is a Raman spectra from the carbonate globule from Boktor volcanic complex. And you see these two characteristic carbon peaks. If you look at the Allen Hills globules in the same textural setting or in the same position within the zonation, we find the same organic chemistry that's quite likely formed on Mars when these globules formed. So we've detected probably the first evidence for organosynthesis on another planet. More interestingly, actually, if you look at interplanetary dust particles, they have the exact same Raman signature as these carbonate globules with their organic chemistry. Well, we shouldn't draw conclusions too far, but we can try. If these things really form by rapid freezing, you might get close, because that's maybe also a process which was involved in formation of these IDPs and cometary material, for that sake. Rapid freezing in a solar nebula, for example. So if we can use the Svalbard data to actually say that we have proof that organic chemistry forms during rapid freezing, you have a kind of mechanism that's certainly present elsewhere in the universe. So we have these other strange features on Sverige, which are these vents, one of them here. And to start with, we found down at the shore, actually, we found these large chunks of carbonate, massive carbonate. And we didn't know where they came from, so we went just zigzagged up the side of the volcano until we came to a place where you had this ring of massive carbonate standing up through the gravel. And we started digging, and we unearthed fantastic vents which is coated with carbonate deposits. And they have the same chemistry and the same carbon isotope composition, the carbon and oxygen, as these globules. So they probably formed by the same mechanism. And by chance, well, first year we found them, we sampled them, and then by chance we continued digging. And about 1 meter down we hit blue ice, clear blue ice, which apparently is water that was sitting in these pipes associated with these carbonates, and that froze in before you started to fill gravel on top. And any hole like that on a volcano that's undergoing frost weathering and stuff like that will be filled with gravel almost immediately. So this ice is probably, you can't prove it and you can't date ice, but it's probably about as old as the volcano itself, possibly up to 1 million years old. So then, of course, we think we have a sample of the water that actually was involved in forming these carbonates. And last year we went out and bought an ice corer and did lots of tests on the nearby glaciers to figure out how on earth you sterilize an instrument like that. So with some work we figured out a way to sterilize it, and then we carefully opened up one of these vents and we took a core into that ice. Unfortunately, these pipes are not straight down, so they curve a little bit, and it took about 30 centimeters before I hit the side, so the core is not very long. Here it is. But it's sufficiently long to be a success. So, you see it's totally clear without any sort of large rock debris in it, but there are certain layers where you can see these essentially clay minerals and small carbonate fragments, which are probably surfaces where the thing was frozen and stuff fell down, and then you got more water on top, and it froze and stuff fell down. So you captured debris while this ice slowly formed. And so we took the sample sterilely, and we were able then to analyze it directly in the field. And this is using the LAL instrument, the horseshoe crab device. And so this is a count of sort of number of or response to cells. You can see the top of the ice, of course, there's lots of activity. And on the outside of the core, there's a good deal of activity. Now, more important that the coring device was more or less totally clean. But you see that even inside the ice, this brown stuff, and inside the core hole itself, there are cells present. Then we use the ATP instrument to sort of count the cellular activity. And again, you see inside the ice, top of the ice, I mean almost everywhere, there are cells, live cells present. And nothing is blank as well. Then we did PCR on extracts from the ice, both from the bottom of the core itself and from this green stuff inside the ice. And we found that we had both bacteria and likely archaea in there, but more surprisingly, cyanobacteria. Cyanobacteria are photosynthetic. And what on earth is a photosynthetic organism doing in the dark beneath 1 meter of gravel? So this is probably an ecosystem that's clinging to these small impurities of tiny mineral fragments, and have been clinging to them ever since they fell into that hole, probably hundreds of thousands of years ago. And more interestingly, they are actually alive. They're not doing much, and they're probably sitting there and thinking, oh, it's cold. But they are alive. And this kind of setting where you have ice under the surface is also a very sort of good Mars analog. And God knows how long these bacteria actually could survive in a setting like that. But it's certainly an example of the kind of organisms you should be able to detect if you ever manage to send something to Mars. Now for some brief talk about future plans. We've got funding at least through 2008 to test MSL instruments, hopefully more than the ones we started with this year. The GCMS, the XRD, XRF, and the sample handling system. And there's also other instruments like the Raman laser and that we hope to get on board the project. So the goal is to establish these localities on Svalbard as a sort of common test site for payload, and to develop protocols for sampling that are common not only for NASA missions but also for ESA. So hopefully from next year we will also have ESA instruments on board. And all these outcrops are now very, very well studied, I mean in all ways and shapes, so we know what we should find with the instruments. And they are an ideal place to test various types of questions you want to ask if you ever get to send them to another planet. So I think I'll stop there. There's lots of other sites too, but I'll leave that for the next talk.
Thank you, Hans.
Yes.